Okay, so last but not least, we have Dr. Steven Kemp talking for us. He's doing a talk titled Community-Acquired Extended Spectrum Beta-lactamase Resistance in Kenyan Patients. So Steve is a postdoc affiliate with Trinity College and holds a research associate position in Ravindra Gupta's lab as part of the Cambridge Institute of Therapeutic Immunology and Infectious Diseases within the Department of Medicine. He has published over 20 papers on the topic of SARS-CoV-2 spike protein and infectivity, as well as the evolution of COVID during chronic infection. So without further ado, Steve, the stage is yours. Thank you.
A round of applause. I was only joking. Thank you, though. Right, let's see how this works. Can we click? Can we type? Yeah, it's because I messed around with the graphics. Okay, cool. The perceptive amongst you might have noticed I changed the title ever so slightly because I realized I wasn't really talking about patients. I was more talking about AMR in a One Health sort of aspect, but we've got some patients in there. So let me tell you a little bit about this study. The screen is really tiny as well. I need to get glasses out. So AMR in this setting, like Dame Sally actually said this morning, around 80% of the antimicrobials used are for livestock production, making sure they're healthy and making sure people can enjoy those animal products they have for breakfast, lunch, and dinner. So obviously we know that antimicrobials are very essential for maintaining both human and animal health. There's a load of different factors related to antimicrobials which will generate either emergence, transmission, or persistence of AMR within these populations. So a couple of those things might be overuse, underuse, using expired drugs, using inappropriate drugs, self-treating when it's not actually required. Yeah, lots of different things. So in Kenya, which is where this study was conducted, there's actually a big shift towards intensification and commercialization of animal products. So like 9 in 10 people keep some sort of animals. That could be cattle, could be pigs, could be poultry, all for the purpose of making a bit of money, essentially. So those livestock are actually a big reservoir of AMR bacteria. And then the slaughterhouse workers who we also investigated are also likely to be a reservoir of the AMR bacteria by way of interacting with those carcasses and slaughtering them and touching poop and things like that. What's really cool that I find is that out of the major 14 antimicrobial classes that we have, 9 of those, which the WHO considers to be critically important for human health, are used in livestock as well. Like identical drugs are used in livestock and human health. So one step above the critically important antimicrobials for human health are these ones termed HPCIAs, which again, Dame Sally spoke about this morning. So these are the highest priority critically important antimicrobials for human health. If you're in hospital and you're this close from death, you're going to get one of these drugs of last resort, as they're called. So these include 3rd, 4th, I think we have 5th generation cephalosporins now as well. Fluoroquinolones and polymyxins, things like colistin. Very useful for human health, but very poisonous. So not really the best thing that you want to be taking, but you know, if it'll save your life, good for you. Obviously, if you're giving a lot of these antimicrobials to your livestock, there's always that argument that they are the major reservoir of AMR, and it's likely that there'll be transmission of bacteria from the animals to humans. Now, that is really rather difficult to quantify. Mostly because, well, I'll talk a little bit about studies in a little while, but if you're giving 80% of the antimicrobials to livestock, are you telling me that 80% of transmission of resistant bacteria is coming from animals to humans? No, definitely not the answer. So a lot of different studies, they basically say, if there's 2 very similar strains of a specific bacterium, like if we're finding one in my cow and one in my pig, we sequence it and we say, quite similar. Usually a study will say there's been direct transmission between the 2. But again, that's not always the case because they failed to consider there might have been sharing from, say, for instance, my cow to the ground to my pig, or from the ground, I don't know, backwards, forwards. There could be a human in the middle. It's very hard to disaggregate. So this study, like I said, is considering it from a One Health, AMR from a One Health perspective. So we're considering human health, animal health, and the environmental health. I've broadly split this study into these different points. So long story short, first of all, we needed to sort of figure out, how do people access antimicrobials? What do people know about AMR? Are they following good prescription practices? Once we did that, then we started to collect fecal samples to basically isolate E. coli from animal poo, human poo, and then we used things called boot swabs to collect environmental samples. So after we've collected all those samples, we then wanted to basically analyze the prevalence of AMR in both slaughterhouse workers and our farm cohort. I then wanted to look at the different circulating AMR genes and sort of correlate that with phenotypic and genotypic resistance. And then I basically wanted to do what other studies have done, which is look at this similarity in bacteria between different hosts, and then also to go one step further and actually say, this is the direction the bacteria has traveled from. And we do that using some cool new tools that we developed over the last 3 years during COVID. We use E. coli mostly because it's very comprehensively studied. We know a lot about it, easy to grow in the lab, easy to genetically manipulate. And basically, if you take any sort of antibiotic that's absorbed by the GI tract, it's pretty likely your E. coli is going to pick up some sort of resistance gene. It's also a major contaminant of carcasses, so it's quite relevant for our slaughterhouse study. So our CAP study, knowledge, attitudes, and practices. Some relevant background that you probably need to know to understand this Kenyan cohort is that basically all of your drugs, or veterinary drugs rather, are bought from these shops called agrovet shops. So agrovet shops sell veterinary antimicrobials, and they also sell, things like rakes, soil, anything else you need to grow crops. Human drugs, you get them from pharmacies, or as our colleague said, you can also get them from the man wandering around with the basket on his head. Kenya and Ghana share quite a lot of similarities. Agrovets typically get staffed by a number of different people. It could be your son who's just come home from school and he has been instructed to sell whatever the customer asks for. It could be someone who's been to high school. It could be someone who's been to secondary school. It could be some random waif off the street. It's very variable. On the flip side of that, you have these things called animal healthcare assistants. So animal healthcare assistants are more likely to have some sort of college diploma or college certificate, which means they have some sort of knowledge and understanding of these drugs, and they can not legally prescribe them, but depending on who you ask, they may or may not be able to legally sell them. To prescribe them, you now need a pharmacist or a vet, like who's got a veterinary degree. In reality, this doesn't happen at all. You can buy whatever you want from whoever you want, whenever you want. So there's a lot of inappropriate selling and prescription. So we used a structured questionnaire basically to interview all of these people. The study site looks like this. So we conducted the study in Western Kenya. So you can see in the middle, do I have a pointer? There's no pointer. In the middle, there's like a little red outline, and then blown up is this on the right-hand side. So this county in Western Kenya is broadly split into like 7 different sub-counties. So in order to get like sort of good geospatial distribution and see if there's any different patterns between different places, we tried to spread out the farms as much as we could. We then tried to interview literally every single veterinary professional that would come, and didn't require a massive bribe for us to talk to them. It did happen often, so we didn't get as many as we wanted to. And then again, agrovet shops. There's not a lot of white people in this area. So when you have random white guy walking around with a clipboard, you tend to find most of these shops closed because they think that I'm an inspector. So again, despite the fact we had loads of farms, we didn't get that many agrovet shops because most would shut up shop when they saw us coming, which is kind of sad. But having seen what we've seen, I can understand why. So here's some just very basic cohort demographics. You can see gender, age group. Job position is what I was referring to earlier. So these agrovets, which is the first 2 columns, you can see that most of them were just manned by like agrovet assistants. Some of them were just manned by the owner of the business. One was managed by a laboratory staff technician who was actually working in our lab. I didn't realize he also owned a business that we interviewed. That was quite cool. and then one of them was like an animal healthcare worker. The most important part of this slide really is to look at the different sorts of education that the farmers have and the veterinary professionals have. So farmers typically, if you are going to make your life's work being a farmer, you don't really need university education. So most of them have either primary school education or a secondary school education. What's a bit more dodge, a bit more suspect, is that our animal healthcare assistants and our agrovet staff in particular, are not really that well educated. So they have, well, more than half of our agrovet staff only have secondary school education. So now imagine you're going now into our pharmacy and you see this gentleman here and you say, oh, what sort of degree have you got? And he goes, I went to school till I was 14, that was it. And you're buying your drugs from that person. That is the reality of what usually happens in these shops in Kenya. Very few of them have a university education or really know much about the antibiotics they're selling beyond what they can find on the packets. So the most commonly sold antimicrobials, overwhelmingly, you usually only find 2 different antimicrobials being either asked for, prescribed, or sold. These include oxytetracyclines and penicillin streptomycins, which are relatively broad spectrum empirical therapies. There is a lack of diagnostics in this area because it's a very expensive thing to do. And, excuse me, most farmers will, A, not be able to afford it, and B, there's not actually that many diagnostic staff in the entire area who could man a laboratory if there was one there. Veterinary professionals also likes oxytetracycline, penicillin, streptomycin. They also quite like sulfur drugs. And then I've just put a little arrow next to gentamicin down there because veterinary professionals who had a veterinary degree would often resort to using gentamicin if nothing else worked. This is a really cool point. So selling drugs to farmers is all about money. You're not really caring if there's a prescription. You're not really caring if you don't really care about anything actually basically besides money. There is a stark difference between the agrovets who are selling the drugs and the veterinary professionals that want to use the drugs to actually treat the animals. So agrovets, if I don't sell my drugs, I'm not making any money. Animal healthcare assistants, if I'm not using those drugs that you bought to treat your animals for you, I'm not making money. Agrovets will therefore tell you as much as you want about the drugs so that they can sell more drugs to you. Whereas overwhelmingly, the veterinary professionals will try to tell you as little as possible because they want you to remain uninformed so that they can charge you for treating the animals. So now that we know what sort of drugs are being purchased, we then want to know, do you really understand what AMR is? So we ask this question, what do you think is the main cause of AMR? And not really that many people know. I find it really interesting here that only about 30% of our farmers said they didn't know. The farmers were much more likely to take a guess at what AMR was, mostly because if they don't want to pay for the services of an agrovet or they don't want to pay for the services of a veterinary professional, they basically just use Google like a smart person. Agrovets just look blank and say, I don't know, I just sell what my boss tells me to sell, and don't really understand about what causes AMR. What's also a bit suspect is that obviously I mentioned intensification and commercialization. If you are treating your animals for a specific disease, you obviously have something called a withdrawal period. So if I give my chickens, chloramphenicol, whatever, any of the eggs that they make or any of their meat will then have some sort of residue, antimicrobial residue. So you legally should wait a certain period, depending on what drug it is, so that all the residues basically exit that chicken. In reality, if you're relying on, actually I think cow milk was probably better used. Say for instance, your cow is being milked right now. If you've just treated that cow, then there's going to be tetracycline, chloramphenicol, whatever residue in the milk. If you rely on selling that milk to feed your family, are you really going to wait for the withdrawal period to end? Bearing in mind the withdrawal period could be 7 to 10 days. No, most of them still sell or they give it to their animals, like dogs. Guard dogs is quite common in Kenya. They'll give it to them so they don't waste any products. So essentially speaking, you can get whatever you want whenever you want. I never in my entire period out there saw a physical prescription. Sometimes during these studies, they just kind of tell you what you want to hear so that they don't get in trouble. What I thought was quite interesting is that around 60% of the veterinary professionals said they did provide a prescription. And then independently, again, around 60% of agri-vets said they dispensed antibiotics against a prescription. But I can tell you, hand on heart, there's no prescription in Kenya. You buy what you want when you want. There wasn't actually any confirmed cases of AMR, like I mentioned, because there's not that much diagnostic capacity. Almost nobody had heard of the term HPCIA before. And then most of the veterinary professionals were completely unaware that there were any sort of guidelines in place. So there's no, the Kenyan government does provide a cascade system. So basically you can very quickly have a look and you can say, right, my patient has come in and they have this symptom, so I should maybe try this or I should ask this. That framework does exist. It was actually set up by the Fleming Fund, but no one is aware of it. So like I said, there's quite a lot of over-reliance on broad spectrum therapies. Only a few people actually reported that they'd had some sort of issue with treating their animals. So around 10% of people said that oxytetracyclines had not worked, around 4% said penicillin streptomycin didn't, and around 8% said sulfonamides didn't work. Again, here's some more lies. I used to work at the only diagnostic lab in the area. That was where our base was. Where there is cases of treatment failure, all of the veterinary professionals said, well, half of the veterinary professionals said that they would take basically a venous blood sample. And then like you do in high school science, you make like a smear on a glass plate, glass slide, and then you look at trypanosomes under the microscope. That's what it should look like. So you can very quickly say, oh, there's something in here I can use to treat this. Again, no one did that because I worked at the only lab where that would have been processed and we didn't get one sample come in. And we also traveled with a lot of them as well. And I never once saw anybody collect any sample, neither did any of my colleagues. So theoretically speaking, if there's some sort of failure with your treatment, you would just switch to another drug. There'd be no diagnostics, there'd be very few questions. You would just go back and say, I bought this from you yesterday, what can I do now? And they'll say, you've tried X, now try Y. And then if for some reason your animal's still alive and still ill, you'd go back and try Z instead. Obviously, we also had to look at the humans, not just animals. Out of all of those humans that we interviewed, so I said 70, but really we interviewed entire families, so there was a lot more than 70. There seemed to be quite a lot of diarrhea and GI bleeding, nausea, mostly a lot of gastrointestinal stuff. Because if you're living with a lot of animals on your farm, for example, sanitation is not always 100%. So out of all of those people who got ill, 91% of them thought it'd be a good idea to get some antibiotics. Didn't just wait for it to pass, didn't just try, supportive therapies, just went straight for hard drugs. And you can buy, any of these in any pharmacy in Kenya. Metronidazole was one of the most common ones. Ciprofloxacin's there, Augmentin. In Kenya, we can actually get hold of it sometimes. And then you have a couple of the first generation cephalosporins. So in summary, from that knowledge, attitudes and practice study, we can see that not many people know what AMR is, not many people are aware of the risks of antimicrobial use, quite a lot of inappropriate prescribing practices, very little diagnostics and surveillance of AMR. Well, there's a little bit more now that the Fleming Fund has started, but when we did this study 5-6 years ago, there wasn't really much going on. And then quite a strong over-reliance on broad spectrum empirical therapies. So let's have a quick look at the prevalence of AMR that we find. So we collected fecal samples, or we collected water samples, or we collected those boot swab samples. We isolated E. coli from them using this McConkey agar. McConkey agar is a pink agar, which is basically really good for picking out gram-negative Enterobacteriales. So they grow as like bright green metallic colonies. So they're really easy to spot. So you don't need to do any other tests, really. We then used very standard microbiological tests to assess AMR. So we looked at a panel of 8 antimicrobials from different classes. I'll show you some of the results on the next page. So we used a disk diffusion assay to type those, and then we also used a double disk diffusion assay to look at extended spectrum beta-lactamases. Sequencing is very, very expensive. So unfortunately, we could not afford to sequence 1,780 samples. So we sent about 400 for sequencing in total. And then from those reconstructed plasmids and looked at transmission. So this is the proportion of phenotypic AMR that we can see in our farm study. Each of the colored bars represents a different antibiotic. The take-home point of this slide is that the green one is tetracycline, the blue one is sulfathiazole, which is a sulfa drug, the red one is trimethoprim, and the dark green one is multi-drug resistance. What did I miss? I missed one of them, whatever. There's basically 4 antibiotic classes that almost everybody is resistant to. So you can see the dashed line going horizontally is about 50%, and then the one down the bottom is 15%. So of those 4 antimicrobial classes, every single cattle, pig, poultry, shoats is sheep and goats, by the way. Humans, the water samples and the environmental samples all had resistance to at least, well, 50% of them had resistance to like 4 or more antimicrobials. When you look at the phenotypes and you break them down, you can see most of them were also multi-drug resistant, usually to tetracycline, sulfonamides, ampicillin, trimethoprim. This is kind of mostly what we saw was offered as most commonly prescribed or sold antimicrobials, but I think there's a lot of underreporting in this cohort. Mostly because when you wander around the farm, you can also see lots of different strewn packets of antibiotics that they have not declared to you. So while they tell you they don't use cephalosporins or they tell you they don't use something else, they often do, because we can see the evidence on the ground. So comparing phenotypic resistance with genotypic resistance, once we've done the sequencing, you can see all of the different resistance genes here. There's various different resistance genes to aminoglycosides, beta-lactams, chloramphenicol, trimethoprim, quinoline, sulfonamides, and tetracyclines. What we start to see here is we have this beta-lactamase gene called BLA-CTXM15. Now, this is one of the extended spectrum beta-lactamases. It's basically a massive public health risk because extended spectrum beta-lactamases are usually co-carried alongside other AMR genes. So where you see ESBLs, you also usually see heavily multi-drug resistant bacterium. We also thought it was quite weird that there seemed to be quite a high degree of environmental contamination with AMR E. coli. So say, for instance, here you can see APH61D on the third from the left. You can see that the environment, every single sample we collected had this resistance gene, which suggests to me that there's a lot of leakage of resistant bacteria into people's water or into people's environment. But this is probably spread by the animals, to be honest. What's also quite cool is that on the left-hand side, you can see the location of all the different farms. And then on the right-hand side, you can see the farms that we managed to isolate extended spectrum beta-lactamase resistant E. coli from. I very crudely drew out for you basically the main major roads in this sub-county, which is in light blue. And then the circle is where our lab was. The circle where our lab was is also where one of the largest agrovets is. It basically serves the entire sub-county. So if people are really struggling with treating the animals, they basically come back to where that circle is along one of these major roads, and then they can get back to their farms using one of those major roads, which kind of explains why most of these extended spectrum beta-lactamase farms are in that sort of central region. So keep ESBLs in mind. We'll come back to them after we look at the slaughterhouse very quickly. this is quite cool because this is the phenotypic resistance that we found in the humans from the slaughterhouses. And you can see on the right-hand side, we now have the humans from the farm study. Whilst the levels of tetracycline, trimethoprim, sulfathiazole, and ampicillin is relatively similar, you can see that the humans that work in the slaughterhouse have much higher prevalence of resistance to these antibiotics. Sorry, these antibiotics they said that they don't take. Now we wonder if that's more reflective of the animals that they're slaughtering. So if we have a look at the resistance genes again very quickly, one of the examples here, so if you remember over here, chloramphenicol was quite low amongst the farmers, but it's around what? It's 15% in farmers, but it's now closer to 40% in the slaughterhouse group. We can see basically a lot more phenicol resistance genes, and this is reflective of people interacting with those carcasses. So similarly, Kenya also banned chloramphenicol and other drugs for growth promotion quite a long time ago, actually. But of course, everybody still uses them because they work. So people that are giving these chloramphenicol drugs, for instance, to the animals, they're then slaughtering them and they're coming into contact with fecal material, organs, things like that containing chloramphenicol residues, and it's contaminating them. And then resistance genes are spreading between the animal E. coli and the human E. coli. The only difference now in resistance phenotypes between the farm study and the slaughterhouse study is that there seem to be many, many more multi-drug resistant and ESBL containing phenotypes in the slaughterhouse cohort. And then also we had some pan-resistant E. coli. So pan-resistant just means completely resistant to every single class of antimicrobial that you test, which is really scary because now if that person gets ill, they're pretty much done for. Right, so we've got ESBL genes, we've got AMR genes, and we've got phenotypic resistance. What I'm now interested in is how are people sharing these things? So they can do that by these things called mobile genetic elements or MGEs, and this is what carries, it's little bits of DNA which carry AMR and ESBL genes. So from the farm cohort, we had 264 whole genome sequences from 264 isolates. We managed to reconstruct 1,172 plasmids from those, and we saw that around 45% of them were either conjugative or mobilizable. So that means that they are able to take this string of DNA out with all these resistance genes on it and give it to something else. So here's an example, a real life example, which is in the paper that we published, of a conjugative plasmid. And this is carrying all these different resistance genes: sulfonamide, aminoglycoside, tetracycline, extended spectrum beta-lactamase, quinolone, trimethoprim. 6 different classes of antimicrobial resistance that thing is carrying. And that's being transmitted between 2 different E. coli or an E. coli and a staph or whatever. From the slaughterhouse cohort, we can now see that we had 157 whole genomes. We managed to reconstruct 618 plasmids, and slightly more of these were either conjugative or mobilizable. And again, here we can see a real life example of one of the plasmids that we managed to find from a mixed ruminant slaughterhouse. This one carried even more. So it's got what? Sulfonamide, aminoglycoside, tetracycline, ESBL, chloramphenicol, quinolone, trimethoprim. 7 different resistance genes. Having known all that, we basically started to get excited about finding all of these BLAST-CTX-M15 genes. So BLAST-CTX-M15 is one of the major ESBL genes. What we did was we looked at geographically distinct isolates from, in this case, humans, the environment, and some animals. And we can see that the resistance genes carried by each of those are identical. The sequence type, identical. The virulence genes, apart from the bottom isolate that had 1 extra virulence gene, also identical. And all of the plasmid replicon types, identical. So this is what we call clonal sharing. It's the exact same or extremely similar bacteria found in one host as in another. So having known that we have clonal sharing, we now want to try and infer who's giving what to who and how is it being done. So as most people do, what we did was we constructed a core genome alignment of all of these sequences. E. coli is between what? 4.8 million to 6.3 million bases long, depending on which reference you use. Trying to align that bioinformatically, using bioinformatic methods is literally impossible. It would take months to do. So what we do is we look to see if I have 400 sequences, which are the genes that I find in 90 or 95% of those samples, and then you basically concatenate them into a much smaller sequence. So we take all of the genes, so what, call it 300 regulatory genes, which takes your genome size now from 6 million bases down to around 200,000. We then align that to a reference sequence, and we say, right, which of the amino acids are different along this sequence? And then all of the small differences are called core genome SNPs, or single nucleotide polymorphisms. So we basically determined that of these 420, 430 sequences I think were left, we could find very closely related pairs that were within 50 SNPs of each other. And then when we started to do some ROC curves and some graphing and some cool stuff with this, we noticed basically a bimodal distribution. So those which were on the same farm or on the same slaughterhouse were actually within 15 SNPs of each other, which shows that they were almost identical bacteria being shared between the two. When you identify those pairs, for example, we saw that rather than going from animal to human or from environment to human or whatever, most of the bacterial sharing was between animals or between the animals and environments. So in the top, you can see all of the different pairs that we basically managed to reconstruct. So you have animal, environment, and human, AEH. Like I said, the majority of them on the left-hand bar, animal to animal. Then you have animal to environment. Then we had human to human, then environment to human, and then finally environment to environment and animal to human. When we start to reconstruct these with phylogenetic trees, phylogenetic tree is basically just a way of showing evolution of bacterial or viral sequences over time. So you would start with a root, which is in the top left-hand corner on the left-hand figure there. Basically, the longer the branch, the more evolution has happened over time. So what I've done is I've just blown this up and basically wanted to highlight some of these very, 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 very similar sequences. So here you can basically see an exactly straight line, which means there's no difference between any of those sequences. They're completely identical, but they were all isolated from geographically distinct farms. And from different, well, you've got environment there, you've got animals and three different animals. They all have the same sequence type. Most of them have the same serotype, and all of them are carrying the same BLA-CTXM ESPL gene. In the bottom there, you can see kind of the same sorts of patterns. They're from geographically distinct farms. They all have the same sequence type, 196. They all have the same serotype, apart from that one in the middle, which is a bit dodgy, which is 08H7. And again, they're all carrying the same ESBL gene, BLA-CTXM15. Oops, too far. When you look at the slaughterhouse cohort, this one was a bit more difficult to disaggregate. So again, we did the same sort of thing. We looked at multi-locus sequence type, we looked at the serotyping, and then we looked at the different ESBL genes that they were carrying. So all of these were from geographically distinct slaughterhouses. So they could have been 40 kilometers from each other, but they all cluster quite nicely together. Most of them have similar sequence types. So 361, 617. What else do we have? We had 10, there was 13. We had some ST131, which is the pandemic lineage. Yeah, we have the clusters again of the same sort of serotype groups. When they get interspersed, it's likely because there was either some contamination or because there was just something that had gone wrong with the program. And usually I would cut those out, to be honest. What was interesting in the slaughterhouse group is that you now have, rather than BLA-CTXM15, we now have BLA-SHIV12, which is a different sort, well, it's basically just a completely different class of extended spectrum beta-lactamase that still carries all those extra drug resistance mutations. So we just have a little bit of ongoing work. So we're in the process of writing a manuscript for another paper. Basically, we see that there's a high degree of circulating AMR amongst all these E. coli isolates. There's obviously very strong evidence, which is supported by statistics of bacterial sharing between 3 groups. So that's animals, humans, environments. There's a lot of ESBR resistance genes which are circulating in this population, and they are on either mobilizable or conjugative plasmids, which means that there is a risk that they will be transmitted. So what we're doing differently to other people is basically using a combination of dated phylogenies, looking at patristic distance of those sequences, and also overlaying all of the different epidemiological data so that I can say, you have given it to you, rather than just saying you both have the same sort of bacteria, so one of you must have given it to each other. And that's what's novel about this study. And then once we've done that, once we've managed to basically say you have given this to you, we can do things like investigate the different resistance genes on both of those genomes, and we can say for certain, have you transmitted everything, or have you transmitted partial? Have you only transmitted one plasmid? What's going on? This is quite cool because it challenges certain other literature that's been published recently, which suggests that ESBRs are actually encoded endogenously on the chromosome in nosocomial settings. But obviously we're looking in a community setting, and we're seeing that half of our plasmids are mobilizable or conjugative, and they're not encoded endogenously. So we wonder if there's some sort of difference between community and nosocomial settings, and we'll explore that a little bit more in the future. So there's some acknowledgements from all the different people that have put some money into this project. I think that's pretty much me. I'm going to take a big breath now and answer any questions you have. Thank you. Of course. They're not very expensive, and because they're not that widely known, farmers will buy them if you try and peddle it to them. So yes, there was evidence. And no. CTXM15, no. Shiv14, no. CTXM14, yes. The ones that I've shown here is not the complete set because there's obviously loads and loads and loads of them. Some of them do, some of them do not. But there's not a lot of pressure for them to be expressed. They are used, but not widely at all. Cool. Thank you.